we can see that here you can appreciate the CD15 positive neutrophil, and with the DNA staidy coming out and basically binding it here in the Shigella. So we can see that both in cultures alone and in co-culture. And in fact, some of these DNA nets can extend many, many microns away from the body of the neutrophil. Just to also show again that we could demonstrate that there is specific recognition of neutrophils to pathogens versus, in this case, an isogenic strain of Shigella. Hopefully what you can appreciate is the number of neutrophils transmigrating through the epithelium, again, increasing the presence of the active virulent strain 2457T, but in the isogenic strain BS103, there was no significant difference either in the number of neutrophils that transmigrated across the epithelium. Another thing that Jose found, which is really interesting, and was building off of data that we also found in the macrophage work, was that as we put these primary cell types together, we're able to find that a synergism could be generated between the cell types. And what Jose did was he did cell surface marker expression of a number of different neutrophil markers and found that based on how the cold culture system was set up, whether neutrophils were either in direct contact with the epithelium or in close opposition to the epithelium versus being at a distance farther away, actually affected the surface expression, surface marker expression on the neutrophils, suggesting that the direct contact or the intimate contact actually affects. And it gave us a number of different phenotypes where we could see that in co-culture, there's a benefit in increased marker expression of CD18, 47, and CD88 in expression on these neutrophils in co-culture, which you can see is significantly upregulated over those that were isolated from peripheral blood. So we saw that as a phenotype. We also found that there were markers that were only specifically upregulated when they were when the neutrophils were in direct contact with the epithelium, versus those where we could see that there's a different phenotype for those that were separated away. And finally, we could also show using this system that the cell surface marker expression could further be altered in the presence of Shigella. And this is just demonstrating that markers of activation of neutrophils in relation to phagocytosis and migration were all upregulated in the presence of Shigella. So another way for us to go beyond just looking at the epithelium, just from the context of looking at crypts and villi, we know that the Peyer's patch is a very most important site for induction of the immune system within the gut, since the gut's basically the largest immune organ in the body. The number of these Peyer's patches numbers in the tens of thousands in the human gut. And so we've been very interested in understanding, can we start to learn about human microfold cells, which is the predominant sampling cell that exists in the Peyer's patch. And this is very important for us from a host microbe standpoint because this is how the gut epithelium samples pathogens, directs them to the immune system, to the follicle in the Peyer's patch, and really drives the initiation of the preliminary immune response at this inductive site. And as these cells are introduced and are developed recognition, then they disseminate throughout the gut to basically create the function of the barrier down the road. These cells are highly unique in the fact that they express a number of pattern recognition receptors that allow them to be very promiscuous in their ability to sample different types of microbes, whether they're parasites, bacteria, or viruses, and also for the fact that they kind of serve as like a parking garage for innate immune cells. So what they do is they have this basal lateral pocket that can serve as the garage, and the different types of immune cells can come in, directly interact with the microfold cells, and capture what is being taken up at the surface and delivered directly to the immune cells. So how can this be done? So I talked to you that in these culture systems, we can drive differentiation of all different types of epithelial cell types, but we can also do that as well within our cells to drive differentiation of M cells by adding an external stromal factor known as rank ligand. And when we do this, and we know from work that was done by others in the field, that the intestinal crypts, the intestinal stem cells, are the source of microfold cells. And hopefully what you can appreciate here, what the LAC-Z staining is that cells that are traced at the base of the crypt eventually become microfold cells over the Peyer's pouch. And so this is also work that Janet has been doing the last several years. So not only can we differentiate to get all the other mature epithelial cell types, which I talked about before, but we can also differentiate these monolayers to express M cells. And hopefully what you can appreciate on the confocal microscopy, in the red staining GP2 is the marker of the mature M cell. We can drive the differentiation of these cell types and reach numbers that we know exist in the normal Peyer's pouch. More so, not only are we driving M cells, we're also driving expression of other follicle-associated epithelial markers. And this is just a demonstration of that both by qPCR as well as by confocal microscopy, showing that we're not just generating one cell type, we're actually affecting the entire epithelium to more recapitulate the follicle-associated epithelium of the Peyer's pouch. So in work that's we did with, in collaboration with Eileen Berry, this is work they published in 2019, and we used this co-culture system and this technique to demonstrate that when I mentioned before that Shigella typically does not invade normal enterocytes through the apical membrane, but utilizes the M cells, in monolayers that were exposed, that were differentiated to express M cells, Eileen's lab was able to demonstrate a significant upregulation in transcytosis of Shigella across these epithelial monolayers. We've also used this same co-culture system to investigate further other invasive pathogens using M cells. And what we've done is we can put fluorescent beads on. We can use this as basically a benign marker to measure transcytosis going through these M cell expressing monolayers. And hopefully what you can appreciate is in the GP2 positive cells, we can see the presence of these beads being internalized. At the same time, these beads also co-localize with GP2, suggesting that they are coming from the apical membrane inside. When we investigated and we tested the hypothesis of whether M cells also have the ability to transcytose and discriminate between pathogens and commensals, we first started looking at enteropathogenic E. coli, which is has known tropism for human M cells, but is not an invasive pathogen. So we wondered if whether or not it could actually induce transcytotic function by just being associated with enteropathogenic E. coli. And what we showed in our monolayer system, measuring the number of beads going across through the pores, we could see whether we were using live bacteria, sonicated bacteria, or LPS from EPEC. We found that LPS did not stimulate transcytosis of the beads going across our M cell monolayers, but EPEC, whether it was alive or dead, could. And also the M cells would only respond to the enteropathogenic E. coli and not the commensal E. coli strain HS. We did some quantitative proteomics analysis. We're very interested in understanding what could be driving this function in human M cells. We know of what a lot of other factors do in the stroma that affects M cell differentiation, but we don't understand what regulates the true machinery within the human M cell that drives this function of transcytosis, which is most critical function. And we ended up discovering the presence of an immune cell factor that has never been shown to be in an epithelial cell. We found it highly expressed in our system. We thought that perhaps this was wrong, but we were able to validate this, that this actin regulating protein coronin 1A was highly expressed and enriched when we differentiated our cultures to express M cells. We validated this in human Peyer's pouch tissue in the follicle-associated epithelium, where we could see coronin 1A expressing cells that were either coronin 1A alone or overlapped with GP2 positive cells. And we could see this very nicely in other samples where, again, as I mentioned, coronin 1A is an immune cell associated gene, and we can see that present in the immune cells. But in the GP2 positive M cells, we also could see coronin in the immune cells that, again, are intercalated into that basolateral pocket. But you can also see in M cells that did not associate with the microfold cells that coronin 1A was there as well. So in an unbiased approach with our colleagues at the University of North Carolina, Scott Magnus's lab, they published this beautiful gut atlas a couple years ago. And in their gut atlas, they also captured follicle-associated epithelial cells from their patients. And we asked them if whether or not our protein coronin 1A was also present in human FAE, and in fact it was. So this was a nice way to validate our work. In order to test and try to understand what the function of this protein is, we knocked it out Again, with these organoid systems, we can genetically alter them either by shRNA knockdown strategies or by CRISPR-Cas9. So we knocked out Corona 1A. This had no effect on any of the paralogs. And it also importantly did not impact the transcription factors SOX8 and SPI-B, which are the critical transcription factors that drive in cell differentiation. And we saw that there was no disruption of either of those transcription factors by mRNA or by protein. And in order to test whether or not Corona 1A is playing a role in M cell function, we went back to our EPEC-treated M cell monolayers, and again, measuring transcytosis, we found that while EPEC was able to stimulate transcytosis in the non-silencing cells, the shRNA knockdown cells completely lost their ability to recognize EPEC and then induce transcytosis. We looked at other cargoes, something more relevant than just fluorescent beads. So we looked at using a chimera construct of the neonatal FC receptor, looking at IgG transport, and with a tool that was generated by Wayne Lentzer's laboratory. And we found that we could see really nice apical to basolateral transcytosis of the FcRn receptor going across in our M cell cultures. But again, our Corona 1A could block that effect as well. But again, looking at IgG and back to our understanding of invasive pathogens, we decided to look at another strain of invasive E. coli. This time we looked at adherent invasive E. coli, which we know not only has tropism for M cells, but can also transcytose. So we performed these infection studies on monolayers for our M cells, and we could see that AIEC is able to transcytose through our M cell expressing monolayers, again, while the HS commensal strain does not. AIEC is able to transcytose without affecting the barrier function, and over the course of infection, we saw no significant decrease in the barrier integrity across these monolayers. And when we knocked down Corona 1A, we could completely block live bacteria transcytosing across these monolayers. And I'm going to leave you finally with one more co-culture system that we've developed, again, using the follicle-associated epithelium M cell expressing monolayers with primary macrophages. And the reason why we decided to do this is because the work of the Loulard lab in France over the last few years has suggested that the innate immune cell populations that exist within the subepithelial dome of the Peyer's patch have a much more increased phagocytic phenotype than the same cells that exist in the neighboring villi. So we wondered if whether or not we could recapitulate again back to this concept that cell-cell interactions can generate a synergy in vitro. We asked whether or not we could see a change in a conversion of the phenotype of these macrophages in co-culture with just having these differentiated M cell cultures. And what we were able to find, either in co-culture or giving macrophages conditioned media for monolayers, we found that we could see a high increased upregulation of lysozyme when these primary monocyte-associated monocyte-derived macrophages are in co-culture versus those when they're expressed in the differentiated cultures. And again, we also looked at Corona 1A because Corona 1A is a protein involved in phagocytosis, and we could see that in co-culture alone, the expression of Corona 1A in these primary macrophages could also go up. And when we looked back at, again, this synergism and this physical interaction between macrophages on one side of the transwall versus the epithelium, when we looked at our co-culture system with M cells, we found that the number of projections just under baseline conditions with these high lysozyme expressing macrophages were just as high as our other co-cultures in which they were differentiated cultures with macrophages exposed to a pathogen. So again, this supports the concept that the epithelium and innate immune cells, particularly the epithelium of the follicle-associated epithelium, creates a microenvironment that allows these innate immune cells to potentially be much more phagocytically active. We're in the process of now taking these to see how does each of these immune cell populations affect the function in the follicle-associated epithelium. So hopefully what I've been able to convince you of today is that bacterial infection with our macrophage-derived co-culture systems work by basically creating almost a clinical model of self-limiting enteritis. by which most of these enteric pathogens, they invade, our body just sort of deals with it. The best thing we can do is just keep our patients dehydrated. And so basically macrophages serve as a first line defense, can clear luminal bacteria within a short period of time. Neutrophils are a little bit more detrimental to the epithelium in the fact they can create weak areas within the epithelium, which makes sense with some of the more chronic inflammatory diseases. But we're able to model a number of different phenotypes that we know takes place with macrophages, including transmigration and mitosis. And also we know that we can drive the differentiation of all the different cell types in our human enteroid modelers, particularly to recreate the follicle-associated epithelium of the Cowries patch, to really now have another in vitro model system that will allow us to look at tropism and invasion of all different types of pathogens in vitro. And we've developed protocols and published protocols now on how to co-culture primary human enteroids and colloids with macrophages, neutrophils. The dendritic cell story is nearly done. We're getting ready to submit that, so look out for that in the future. Our M-cell work is also about ready to be submitted, well, actually about ready to go into press. So, so hopefully look out for those as well. So I want to give a lot of thanks to Jim Caper for leading this group, to Marcella Passetti and her laboratory, with Gail and Jose being really strong partners in developing these co-culture systems, partners within the program project, and Lee Berry, who is here, Sri Ganathan, who was a former postdoc. I want to thank Janet Staub for working on developing the co-culture systems, the M-cell work. Really excited about the work that's going to be coming out of our lab at Vanderbilt at some point soon. Particularly Teresa Torres, my graduate student, is here, who's using the M-cell model to understand how, in a model of ileal Crohn's disease, M-cell function is altered in that disease. So with that, I want to thank everybody, my funding sources, and happy to take any other questions.